This video is going to cover the first part of the cell, which is the cell membrane, and from there we're going to look at other parts of the cell. Remember that cells are the basic unit of life. They need to maintain homeostasis, which is really why the cell membrane is there. They maintain a separation from the environment around them through this, what we call a plasma membrane. Plasma membrane is primarily made of phospholipids, which is this phosphate, a polar part of a molecule, hydrophilic and hydrophobic tails. If you looked very closely, you'd be able to see carbon and hydrogen chains. These carbon and hydrogen chains are going to arrange themselves because of the nonpolar interactions in the inside of this membrane, and the hydrophilic polar parts are going to be on the outside. That's the basic structure of the phospholipid bilayer. Again, think of the words phosphate. Lipid, we should know by now what lipids are, and then bilayer, two layer. The actual phospholipid bilayer is actually a lot more complicated than just the phosphates and the lipids. As we'll see through this video, the actual components of a cell membrane are things like cholesterol. You hear about the bad aspects of cholesterol, but cholesterol does also help stabilize the cell membrane. We have lots of different proteins. We also have glycolipids. So glyco means carbohydrate. Something with a glyco in it is a carbohydrate with, in this particular case, a lipid. Glycolipid is carbohydrate connected to a lipid. Glycoprotein is carbohydrate connected to a protein. These are other important things in the cell membrane. What we also need to remember is that the cell membrane is selectively permeable, which means it lets some things through. Semi-permeable is another word for it. Some things get through, but not everything. And it's very important that not everything gets through, because a cell needs to keep some molecules inside of it and needs to get some molecules out. That's how it maintains homeostasis. A cell's contents are not the same as the environment around it. A cell's contents are very much different than the surrounding environment, which is why they are alive. And if you will remember, when we talked about the origins of the cell, the membrane is really what keeps the good stuff that helps the cell survive inside of it. Those RNA and DNA and proteins are kept within that cell, which may give that cell an advantage over other cells, and which is what allows evolution to take place. Something very interesting when we're talking about phospholipids or the phospholipid bilayer is that the Archaea, we have three different domains of life, fourth possibly being viruses, but we haven't agreed whether they're alive or not. But the Archaea have a different kind of lipid bilayer than bacteria and eukaryotes, which are the cells with a nucleus in them. Very interesting. That possibly means that the Archaea may have actually gotten their RNA and DNA and proteins surrounded by a different kind of phospholipid bilayer originally, which means they may have evolved independently of other cells. Their cells may have developed not from the same common ancestor as bacteria and eukaryotes. Their bilayer is quite a different structure than the bilayer that our cells have and what bacterial cells have. The only reason they're considered prokaryotes, which is a old designation of life had originally been separated into prokaryotes and eukaryotes. But prokaryotes is made of two very, very different groups. Bacteria are very different than Archaea. Archaea live in very extreme environments. Archaea are very interesting in a lot of ways. Their DNA suggests that our ancestors may have gotten some genes from Archaea, and those genes were transferred to us maybe even before cell membranes formed, although we also know that certain prokaryotes can actually even some eukaryotes can shift DNA from one living thing to another, as we'll see when we study plasmids and some other ways of exchanging genes over the course of this year. What I'm trying to make sure you understand here is that Archaea are very unusual organisms, very understudied too. We know they live in extreme environments. They may be the same sort of life we would see on other planets, which is what's really exciting about them. Very understudied. They have a similarity with eukaryotes because of some of their DNA, but we, eukaryotes and bacteria, have the same sort of a cell layer, while Archaea have something completely different, which is very interesting. As for the bacteria, we did take a look at the bacterial capsule, the outermost part of the bacteria with that cells alive activity. The gram-positive bacteria are ones that would stain purple with something called a gram stain. It's a crystal violet. It's a certain chemical that sticks to this peptidoglycan layer. Remember, glyco, something with glyco is a carbohydrate. Peptid, anything with that, is a protein. Peptidoglycan is protein stuck to carbohydrates in this cell wall. That's what the cell wall is made of. The peptidoglycan layer of gram-positive bacteria is much thicker than the peptidoglycan layer of the gram-negative bacteria. The gram-negative bacteria don't stain purple like the gram-positive is because the crystal violet sticks to the peptidoglycan layer. Here in the gram-negative, there's another layer of membrane that protects the cell wall and means that the crystal violet can't actually reach the cell wall. And so what we see in a gram-negative is you see a regular old cell membrane, the peptidoglycan cell wall. Then we have another layer of proteins another layer of cell membrane on the outside, which keeps this from staining. What I had said is that many antibiotics will work on breaking down this wall. And then once this gets broken down, the cell actually doesn't have as much protection as it used to, and the cell can actually break apart. That's what's very important about the use of antibiotics. Many antibiotics, like penicillin, work very well against gram-positive bacteria. Gram-negative bacteria are harder to break down because that antibiotic might not be able to reach the cell wall in order to break it down. This is why generally gram-negative bacteria are harder to treat than gram-positive bacteria. This is very important because we need to understand the process of antibiotic resistance. This is one of the greatest health effects in the world today that's not reported as much as it should. What happens is bacteria, when they're copying their DNA, there might be a mistake. A mistake in DNA copying is called a mutation. That's what it's talking about here. One of the bacteria undergoes a mutation that gets it to be resistant to antibiotics. Therefore, when someone takes an antibiotic, all of the bacteria that are sensitive to that, they get killed, which means we have a whole lot of open space for this resistant bacterium to take over. That's basically an evolutionary change in the bacteria. This is happening a lot. We have some types of tuberculosis, for example, which are resistant to every antibiotic that you can get from a doctor. If someone gets something called XDR, which is extremely drug-resistant tuberculosis, they actually have to go to a special facility and get a very special antibiotic that they don't give to everyone else because we don't want bacteria becoming resistant to these antibiotics. And the challenge is people get antibiotics prescribed for many different things. We actually also feed antibiotics to animals to make them grow faster. But all of that is encouraging this antibiotic resistance. And that is why many of these diseases that used to be treated with antibiotics are becoming harder and harder to treat because we're getting more and more resistance in different types of bacteria. As I said before, the cell membrane is a semi-permeable barrier. Small things can get through. Water can generally get through without a lot of trouble. CO2, carbon dioxide, oxygen, O2 can diffuse through this membrane fairly easily. Nitrogen can too, although the CK12 assignment will tell you that nitrogen can't diffuse. Nitrogen, N2 is very much like oxygen, O2, or carbon dioxide, CO2. They're all molecules that are very small and they can get through the cell membrane. But the cell membrane is a barrier. Cells need to get certain things in. These little rings, those are carbohydrates. These little blobs, they could be something like a protein. There are bigger things that the cell needs to get through that can't diffuse through this barrier. If you remember, carbohydrates have a lot of OHs on them. Hard to get a polar molecule through a nonpolar membrane. So we have proteins that can help get things through that we want to. And proteins might specifically be for bringing through carbohydrates or specifically for bringing through proteins. Also, a cell needs to pump things out of it. Something like maybe there's too much urea, which is a waste product, or too much of a certain ion, like sodium. Now it has to pump that out. This is something that cells need to do in order to maintain homeostasis. So urea is formed from the breakdown of proteins. It's a waste product. It's not something your body wants to keep inside of it. So your urea has to be shipped out of the cell into your bloodstream and then passed out as urine. What we need to know is the membrane is a barrier. All cells, when they undergo metabolism, molecules are broken down for energy. To maintain homeostasis as those molecules are being broken down, we need to bring in certain molecules, maybe more carbohydrates. We need to excrete certain molecules, maybe urea. Which molecules might be absorbed and excreted? I just told you. Carbohydrates would need to be brought in as an energy source. Urea might have to be excreted. There's a lot of other waste products that build up within the cell that might need to be broken down. In addition, some cells are responsible for making certain materials. If it makes it inside the cell, now it has to pump it to a different part of the body. I'm thinking something like a hormone or a steroid, which our bodies make. They might be made in certain specialized cells, but then they need to be pumped to a different part of the body. Insulin is another great example, which helps our body absorb sugar. Insulin's produced in cells in the pancreas, needs to be exported to the rest of the body, and proteins are responsible for doing that. There are many ways, and we'll look at the different ways that items can get in and out of a cell. They might depend on the size of the molecule, the charge of the molecule. We're also talking about ions here. Some of them require extra energy to get it in and out, and the main goal is homeostasis. Keep the same amount of what I need in the cell, get rid of what I don't need so I have a stable internal environment. What you can see here, oxygen, again, diffuses fairly easily. Water goes back and forth without a tremendous amount of trouble. It is hydrophobic in the middle of the cell, but water is such a small molecule it can slip through. Glucose is much larger and has a harder time getting in and out, which is why we have something like a protein to help that make that possible. Diffusion, you'll see this in chemistry. Diffusion is a term you may have seen before. It's where atoms, ions, or molecules will move from an area of high concentration, where there's lots of it, such as here, to an area of low concentration. So all of this crystal or whatever it is, dissolves. All of those molecules or those ions spread out across the liquid. This may also happen across a barrier. This is an example of a semi-permeable barrier. Maybe it's open to something like water. You have more water on one side, a higher concentration of water on one side than the other. Over time, water will cross that barrier because it wants to spread itself out evenly. This is just something that molecules like to do. This could also apply for a gas, such as carbon dioxide or oxygen. And in some cases, that's fine. In some cases, the water can travel across the membrane and not cause any trouble. However, there are incidents where we might get into problems when water is allowed to cross a barrier without balancing the environment. So what I want to say here, if we had something called a hypertonic solution, which is like a really concentrated salt or sugar solution, if we put a cell in that and water can cross the membrane, this cell is going to shrivel up. This cell is going to shrivel up because the water molecules, which are more concentrated inside the cell, are going to leave the cell. And as they leave the cell, they cause the plant cell or the animal cell to shrivel up. That could be a big problem if you are trying to keep alive. If, however, like You make a salt solution to gargle with when you have a sore throat. That's how you kill the bacteria. You actually cause the bacteria to shrivel up because you've put a very highly concentrated salt solution inside those bacterial cells. There's a lot of water. The water comes rushing out of the cells, and you actually kill the bacteria because they lose so much water they can't continue surviving. And that's how a salt gargle will work on a sore throat. Isotonic solutions is where we have a nice balance between what's inside the cell, whether it be salt or sugars, and what's outside the cell. That means that the water doesn't rush out of the cell, the water doesn't rush into the cell. The water's in a balance with its environment. We call that equilibrium. Something that nature loves is when things are balanced, when heat is balanced, when water content is balanced, when salt content is balanced. That's what nature likes. Nature likes balances. However, life doesn't always necessarily like a balance. In an isotonic solution, life is happy with the balance. But hypertonic, this cell's going to have to find a way of fighting that gradient and getting water back into it if it's going to survive. We also have a hypotonic solution, which has less stuff outside the cell. So maybe like distilled water, pure water, will cause cells to swell up. And actually, it will possibly cause them to swell up to the point that they burst, which is called cytolysis, where a cell breaks. That's called cytolysis. In a plant cell, you can give them pure water. They fill up, they start to swell, but then the cell wall actually keeps them from breaking. So cell walls are very useful for plants. When you give water to a plant, that actually is what helps hold the plant up. These walls become very rigid when they're full. Remember, all of the water on the outside is less. There's less of a concentration of salt and sugar. Inside, there's a lot more salt and sugar. So water is going to rush in to these cases, and that will cause the plant cell to swell up. The cell wall keeps it from breaking, but that gives the stiffness in a plant that allows this plant to stand up, which is also very important. We also have compounds or ions that have charges on them. Very hard to get a charged particle through the hydrophobic part of the membrane, although molecules that are hydrophobic, if they're small and they're hydrophobic, they're allowed to pass through the membrane. But a lot of cells are able to bring other items that have charges on them through, such as ions, thanks to things like transport proteins. Transport proteins will move a charged ion across the cell membrane, but not bring it in contact with those hydrophobic portions. What you need to know here is that maybe an ion will connect with one portion of the protein. As soon as that charge connects, the protein actually changes its shape because of hydrophobic and hydrophilic interactions, which means that the ion can then be passed through the cell membrane. Now, for each molecule that needs to get into a cell, we're going to have different transport proteins because all of this is based on shapes of different molecules. I've talked about the shapes and why they're important. Hopefully, we understand a little bit about the shapes. And that is essential for determining what can go through a membrane at any point. This particular protein might be one for moving carbohydrates through. This could be a protein for moving something else, one of these other things, possibly an ion or something through. Same thing here. So each molecule that needs to come into the cell or needs to come out of the cell is going to have a specialized protein in order to move it through. And remember, that's one of the important aspects of a protein. Proteins are very good at making different shapes and shapes that are necessary for different sorts of work inside of a cell. Very, very important. Also, proteins can interact with a wide variety of different substances because they have that hydrophobic portion, which can work with hydrophobic molecules, and the hydrophilic portion, which can work with charged particles. This is the other advantage that proteins have over other macromolecules. There are even some molecules that will require energy in order to do work. So these are proteins that actually need this adenosine triphosphate, which is a nucleic acid. It's also the main energy storage molecule in a cell. You'll see that these sorts of proteins actually need one of these phosphates to break off of an ATP and join onto the protein molecule, and that changes the shape of the molecule to the point that something like these sodium ions, which are sodiums with their electron or outermost electron missing, can be ejected from the cell because there's maybe too much of a concentration inside, and then these potassium ions can join and be brought into the cell because possibly there's some issue where the cell needs more potassium. This is what the proteins do. They get rid of what, and they take in what they need to receive. Now, there are ways of moving larger molecules as well. A little bit of a look here. So endocytosis is when components such in this case a bacterium are brought in the cell because the cell membrane actually changes. Watch the change in shape. The cell membrane forms a vacuole, one of these membrane-bound spaces within the cell. The bacterium gets pulled into the cell and it may be getting eaten there. What you need to understand is the root word endo means inside. Cytosis refers to the cell. It's a process that a cell does to bring things inside. We have phagocytosis. Phage or phagos means devour or eat in regular English. That's important. We're going to see phage a lot in this particular year of biology. So phagocytosis means eating a cell, in this case, one cell eating another. But really what this cell is doing is changing its cell membrane to enclose a cell that it wants to eat or take in for some other reason. I did mention that mitochondria have their own chromosomes. Mitochondria probably came to live in our cells much through the same way. They were a bacterium that was enclosed by a cell, taken in, and then it just so happened that bacterium was able to produce energy efficiently for the rest of the cell, and that's why they were allowed to stay. We also have pinocytosis, which is kind of another way that a cell might bring something in, although that's more of a case of drinking. It's the cell bringing in a liquid that it may want to utilize for some reason. Phagocytosis is eating. Pinocytosis is basically the same thing as drinking. Here we have a good example of phagocytosis. These are white blood cells eating up a whole bunch of bacteria, or amoebas eating up a whole bunch of bacteria. This is how fast phagocytosis can work. And that's the way that cells get larger particles into their cells, into their bodies. One other thing we have is receptor-mediated endocytosis. One thing about the proteins are often on the outsides of cells. Some of these proteins actually work to grab onto things. And once enough of this protein is grabbing onto something, it will actually pull the substance that it wants to bring inside. It will pull it into the cell as it wants. The particular case here is actually a virus being brought into the cell. Normally a cell would not want to bring a virus into it, but viruses are very good at getting attached to certain receptors in the cell and sending a signal to the cell that, oh, you need to bring me in. Once it gets in, it takes over the cell, it causes the cell to make more copies of itself, and that is how viruses infect cells. Viruses are very sneaky at getting into cells, but this is basically an example of proteins on the cell membrane grabbing onto something that they want or think they want to grab onto, and then bringing it into the cell itself. That could work for something that is important that the cell might need, that's a large molecule or a large package, or it might be something that the cell doesn't need. These receptors are the special proteins that grab on, they pull it in, the cell membrane will then fold around it. In addition to endocytosis, which is pulling in, we also have exocytosis. You may have learned about the exoskeleton, the skeleton on the outside of certain animals, like insects, have an exoskeleton. Well, exocytosis is the process of getting rid of certain things. So if too many waste products build up within a cell, the cell will have to gather these up, possibly in a vacuole or some sort of a vesicle, and then it will excrete it. We talked when we did the cell modeling, we had a secretory vesicle. That's what this is. It's a vacuole. It's a membrane enclosed space which has some molecules the cell wants to get rid of, and then it will take it to the membrane and eject it out of the cell in order to maintain homeostasis, so it doesn't get too much of a buildup of wastes inside. It's very similar to other things that we've seen. Diffusion, oxygen can diffuse into a cell, carbon dioxide can diffuse out of a cell. Facilitated diffusion is where you might have a protein grabbing something on one side, bringing it in, but it's with a concentration gradient. So maybe you have a lot of sodium on the inside and a lot of potassium on the outside, and the cell will work to bring in the potassium and expel the sodium, as we saw. Although that also falls within the realm of active transport. Active transport is where it is actually using energy and the protein is changing its shape in order to get a molecule in or out of the cell. With that, we end, and we will look at the next cell organelle.